Hi, my name is Abby Komar, and today I will be talking about MERS coronavirus. I will be going over the history of the virus, its biological functioning, and the current treatment and control tactics being used to prevent the spread of the virus. On June 13th, 2012, a 60-year-old man from Saudi Arabia was admitted to a hospital with a fever, cough, and shortness of breath. 11 days later, the patient died of progressive respiratory and renal failure. On September 23rd, 2012, the World Health Organization reported 2 cases of acute respiratory syndrome with renal failure associated with a novel coronavirus in 2 patients from the Middle East. Sporadic cases were detected in the Middle East over the following 6 months, including 2 retrospectively cases diagnosed from Jordan in April of 2012. Additional cases were also reported in Europe and Africa among patients with recent travel to the Arabian Peninsula and their close hospital and household contacts. On May 14th, 2013, the International Committee on the Taxonomy of Viruses officially named the novel virus MERS coronavirus, standing for Middle Eastern Respiratory Syndrome coronavirus. Since 2012, 27 countries have reported cases of MERS, including countries in Africa, Europe, Asia, and North America. 80% of these cases have been reported by Saudi Arabia. All cases of human infection with MERS-CoV have been in people who lived in or traveled to the Middle East, or who had close contact with people who acquired the infection in the Middle East. Many isolated outbreaks have occurred within the Middle East. The largest known outbreak of MERS outside of the Arabian Peninsula occurred in the Republic of Korea in 2015 at a multi-center hospital outbreak. The outbreak was associated with a traveler returning home from the Arabian Peninsula. The majority of outbreaks have occurred in healthcare facilities. Human-to-human transmission typically occurs among family members, patients, and healthcare workers. Human infection of MERS-CoV has been linked to contact with dromedary camels or other humans infected with the virus. They believe that dromedary camels are a reservoir and intermediary host of the virus, and that the virus came to humans via a crossover event. Serological evidence suggests that the virus could have existed among dromedary camels in Saudi Arabia as early as the 1980s. The strongest evidence of direct cross-species transmission of MERS-CoV from camels to humans comes from a study reporting the isolation of the virus in a dromedary camel that had a complete genome sequence identical to that of a human strain from a patient who developed MERS after close contact with sick camels that had rhinorrhea. Rhinorrhea is commonly known as a runny nose. Although it seems evident that humans first contracted MERS-CoV from camels, thus making it a zoonotic disease, I'm going to focus on the human aspect of this disease and its impacts. MERS-CoV likely spreads from an infected person's respiratory secretions, such as through coughing. Human-to-human transmission levels are very low. The virus typically spreads to others through very close contact, such as caring for or living with an infected person. In dromedary camels, MERS-CoV replicates in the nasal epithelium. Symptoms for these camels typically result in something similar to a runny nose. In humans, MERS-CoV replicates in the lower respiratory tract, in the bronchiolar and alveolar epithelia. This difference is due to the presence or lack of the host cell receptor DPP4 in certain cells, which is necessary for the virion to bind to the host cell membrane. In humans, DPP4 is absent from the nasal epithelium but present in the lower respiratory tract epithelium. The opposite is true for dromedary camels, thus explaining the difference in virus tropism between the two. MERS-CoV is in the family of coronavirus, which get their name from their shape. Under the electron microscope, the virus particles appear to have crowns. Corona is the Latin word for crown, and thus they have been dubbed coronaviruses. MERS-CoV is a positive-sense single-stranded RNA virus. This means that the RNA genome can be directly translated into proteins. Here we have a diagram of the virus particle itself. MERS-CoV is a membrane-enveloped virus. Inside the membrane is the RNA genome, which is covered in the nucleocapsid or N proteins. Around the rim of the membrane, we can see multiple membrane or M proteins and envelope or E proteins. The most significant protein on the virus is the large spike or S proteins. The virus uses the S proteins to bind to the host cell that it is infecting. Once inside the lower respiratory tract, MERS-CoV utilizes two different entry routes for infecting host cells: membrane fusion and endocytosis. In both, the S protein on the virus binds to the receptor DPP4 on the host cell. Then, for membrane fusion, the virus and host cell membranes fuse together and the RNA genome is released into the cytoplasm of the cell. For endocytosis, an endosomal membrane is formed around the virus and takes the entire virus particle into the cell. Then the membrane vesicle is broken down by pH triggers and the virus RNA genome is released into the cytoplasm. Once the RNA genome is released into the cytoplasm, the ORF1A and B, or open reading frame, genes are translated into proteins, one of which is the RNA-dependent RNA polymerase. The rDRP makes a full-length negative-sense copy of the genome, which is copied again as positive-sense RNA. This is the genome for the new virus particle. The rDRP also makes copies of the genome that are used to make small viral mRNAs. All of this occurs at the host cell's endoplasmic reticulum because its ribosomes are necessary to translate the mRNA into protein. The mRNAs are translated into viral proteins such as S, M, E, and N proteins. The new virus particle assembles at the Golgi apparatus. The virus then exits the host cell and moves on to infect the next cell. Infection symptoms range from asymptomatic to very severe pneumonia with acute respiratory distress syndrome, or ARDS. The disease starts with a fever, cough, sore throat, or shortness of breath, and rapidly progresses to pneumonia, which often requires ventilatory or other organ support. Almost half of people who are infected with MERS develop pneumonia, and less develop ARDS. At least 1/3 of patients also had gastrointestinal symptoms. Approximately 35% of people with MERS die. Most of the people who die had a pre-existing medical condition. These included diabetes, cancer, chronic lung disease, chronic heart disease, chronic kidney disease, and other comorbidities. Symptoms of MERS start to appear around 5 or 6 days after a person is exposed, but this value can range from 2 to 14 days. There is no specific vaccine or treatment currently available for MERS. However, several MERS-CoV specific vaccines are in development. Patients are treated case by case and are given supportive care to help relieve symptoms. The control measures are very similar to the ones being used now to prevent the spread of SARS-CoV-2. Washing hands, keeping surfaces clean, and washing produce all remain as important steps in preventing the spread and contraction of viruses. Additionally, people living in or traveling to the Middle East should avoid contact with sick camels. Unpasteurized camel milk and undercooked camel meat should not be consumed. Testing should be performed among anyone who potentially had contact with a sick person or camel, even if they are asymptomatic. If someone is suspected of having exposure to the virus, they should undergo medical surveillance and quarantine at a healthcare facility or at home until 14 days after the last day of exposure. Air travel should be restricted for laboratory-confirmed cases unless it is necessary to transfer the patient for medical care. Anyone in contact with an infected person should wear the appropriate PPE. In hospital settings, the suspected cases should be isolated from other patients. MERS-CoV does not easily spread from person to person, unlike the current SARS-CoV. Generally, it has been fairly isolated to the Middle East, and outbreaks rarely occur outside of a healthcare facility. Even though the case fatality rate is 35%, the virus does not spread easily and can be contained by quarantining and isolation. This is one of the biggest differences between MERS outbreaks and the current global pandemic with SARS-CoV-2. However, significant research is necessary to learn more about the functioning of MERS-CoV and to develop an adequate vaccine and treatment to prevent the further spread of the virus. I hope you found this presentation interesting and were able to learn more about MERS-CoV. Thank you.